Hello everybody, we have seen a number of videos on pneumonia. All were of very basic level to make it easy for everybody to understand. It is time for us to see what happens microscopically. To make it simple and easy, I have chosen only one microorganism which is known as Streptococcus pneumoniae. It is a famous gram-positive round bacteria known to cause infections including pharyngitis, meningitis, pneumonia, especially lobar pneumonia in human beings, which we are discussing right now. There can be several outcomes, including death, depending upon several factors. Here, we will only discuss the process of infection uh, and assume complete healing through the process of resolution. Resolution means complete clearing of mess created by bacteria and return of lung to normal function. Some of the processes shown in this slide and in next two slides happen in order, while other happen simultaneously with other processes. Infection starts when bacteria make their way to alveoli of lungs. Alveoli always have macrophages which start phagocytosis, means they start killing the bacteria, and if they completely finish the bacterial population that has managed to enter in alveoli of lung, then nothing goes wrong and process finishes. In case if macrophages cannot kill the whole population, then the rest of the bacteria start damaging the host cell, the alveolar cells, by releasing toxins. Hemolysin is a toxin that is produced by and released by some bacteria including Streptococcus pneumoniae, which damages red blood cells in order to release hemoglobin from it, which is used by bacteria as nutrient, as food. Pneumolysin is another toxin that creates holes in host cells, again to release their contents, which again is used by bacteria as food. Hydrogen peroxide is another toxin that is released by Streptococcus pneumoniae that damages the surrounding tissue. Now, what macrophages do in this case, they release cytokines. Cytokine is a protein which is released by macrophages in order to signal the body to ask for more help. Cytokines also send signals to brain to increase body temperature, to cause fever, and it is this process that starts the next process, which is inflammation. Inflammation means tiny blood vessels in and around infected area become dilated and become more permeable, and now allow plasma proteins to move into affected area. Dilated blood vessels also allow extra blood flow, creating redness, heat, and swelling. And in the meantime, neutrophils are deployed, which start phagocytosis again. Alveoli get filled with exudates, and exudates are composed of dead cells, neutrophils, fibrin, lysed blood, especially lysed RBCs, broken down RBCs. It is this lysed blood which gives brownish color to the sputum of infected person. Fibrin comes from fibrinogen, which is a blood plasma protein, and is a part of healing process. Phagocytosis by neutrophils, and if neutrophils manage to finish the bacterial population, then more macrophages are deployed, and towards the end of the process, fibrinolysis begin, means fibrin starts to break down to be absorbed by the interstitial tissue or the lung tissue, and then finally lung assumes or resumes normal function. Let me show you a few sketches and describe the process again. So this is a cross section of a normal alveolus, which is filled with air, and macrophages are always there. These red things are tiny blood vessels that surround the alveoli. This is an infected and inflamed alveolus. The blood vessels are dilated. The alveolar space is filled with exudates, which is composed of fibrin-lysed blood, bacteria, dead cells, macrophages, and neutrophils. Let me show you a longitudinal cross section from this interface here, which should look like this. So this is a blood vessel, these are endothelial cells, this is a normal alveolar space filled with air. This is alveolus is filled with exudates. As a result of cytokine release, the neutrophils are deployed to the area of infection, and they are heavier than the red blood cells. What they do, they push these endothelial cells apart to make their way to the alveolar space here and start the process of phagocytosis. A typical picture of pneumonia, a lobar pneumonia, which is usually caused by Streptococcus pneumoniae. If you take a sample from here and put it under a microscope, the sample should look like this. So this is pleura, vestral pleura. This is the normal lobe, and this is infected lobe. So the alveoli here are filled with air, as opposed to this junk here, which is composed of, in initial stages, fibrin and neutrophils, and towards the end of the process you'll see more macrophages in there rather than neutrophils. Acknowledgements and references, and the lecture is prepared for academyofprofessionals.com. Thank you very much.